Hello everyone. Today we are exploring the next frontier in genetic therapeutics, peptide nucleic acids, or PNAs. While traditional genetic medicine has shown immense potential, it has long been hindered by limitations in delivery and stability. PNAs are engineered molecules designed specifically to solve these challenges, paving the way for a new era of systemic treatment. The journey of PNAs from the lab to the clinic is driven by three key pillars. First is synthetic anatomy, where we replace the natural backbone to create a neutral, indestructible DNA analog. Second is versatile targeting, allowing us to regulate the genome via antigen, antisense, and direct gene editing pathways. Finally, in vivo delivery leverages gamma modifications and PLGA nanoparticles to overcome historical biological roadblocks. To understand why PNAs are revolutionary, we must look at their chemistry. Traditional DNA and RNA have a negatively charged sugar phosphate backbone, which creates electrostatic repulsion between strands. In contrast, PNAs use a neutral N-2-aminoethyl glycan pseudopeptide backbone. This neutral charge, joined by methylene bridges and carbonyl groups, fundamentally changes how the molecule interacts with genetic material. This neutral backbone gives PNA what we call biological invincibility. Due to the absence of electrostatic repulsion, PNA/DNA hybrids are extremely stable. For example, a 6-base PNA/DNA double helix melts at 31 degrees Celsius, while DNA alone melts below 10 degrees Celsius. Furthermore, PNA is invisible to natural enzymes, making them completely resistant to degradation by nucleases and proteases, thus significantly extending their intracellular half-life. PNAs are highly versatile in how they bind to target nucleic acids. They can form a triplex by binding parallel to a DNA strand via Hoogsteen base pairing. They can perform duplex invasion, where homopurine PNAs displace a DNA strand to form a D-loop. Or they can use a tail clamp TCPNA, two PNAs joined by a flexible linker, to create a highly stable anchor on the target. By binding to DNA or mRNA, PNAs can effectively halt the central dogma of biology. In antigen intervention, the PNA binds to the promoter or template strand at the DNA level to stop transcription. In antisense intervention, the PNA binds to the August initiation region of mRNA, sterically blocking the ribosome from translating the protein without needing to rely on RNase H degradation. Beyond simple blocking, PNAs enable sophisticated therapies. In anti-MER therapy, PNAs bind to oncogenic microRNAs to prevent them from interacting with the RISC complex, restoring normal gene expression. Even more impressively, PNAs can act as a gene editing catalyst. A tail clamp PNA invades genomic DNA, creating a helical distortion that recruits the body's own repair pathways to seamlessly insert a donor DNA fragment. Despite their promise, early clinical translation was stalled by three major warnings. First, unmodified PNAs are hydrophobic and prone to aggregation. Second, they suffer from membrane impermeability. They cannot easily cross the lipid bilayer, with efflux times stretching into days. Third, they face rapid physiological clearance, often being flushed through the kidneys almost instantly, with a circulation half-life in rats of only about 3 minutes. The first evidence for intracellular delivery came from conjugating peptide nucleic acids, PNAs, with cationic cell-penetrating peptides, such as penetrant peptides or TATs. While this approach successfully pulled the molecule across the cell membrane, it encountered new bottlenecks. Because most of the drug remained in the lysosomal pathway, large, repeated administrations, 10 to 50 mg/kg, were required for effectiveness, which not only increased the difficulty of production but also limited efficiency. A major breakthrough occurred when experiments showed that systemically delivered PNAs could cross a healthy blood-brain barrier. In a rat study, an unmodified antisense PNA targeting the neurotensin receptor was detected intact in brain tissue 8 hours after injection. This resulted in a $40% knockdown of the target protein, proving that PNAs could reach the central nervous system through systemic delivery. Innovation in chemical structure further solved the solubility issue. By installing a simple mini-PEG chain at the gamma position of the PNA backbone, we introduced hydrophilic properties that eliminate aggregation. This modification also pre-organizes the PNA into a right-handed helix before it even meets the target DNA, unlocking rapid Watson-Crick invasion under physiological conditions. To achieve massive intracellular delivery, we turn to PLGA nanoparticles. PLGA is an FDA-approved, biocompatible polymer. Using a double emulsion process, we can co-encapsulate both the PNA and donor DNA into a single nanocarrier. This protects the cargo from clearance, eliminates the need for complex CPP conjugation, and drives massive cellular uptake with zero reduction in cell viability. These breakthroughs have already achieved peak success in vivo. In a mouse model of beta thalassemia, intravenous injection of PNA via PLGA nanoparticles achieved 4% gene correction, restoring hemoglobin levels to normal and reversing splenomegaly. In HIV-1 research, this platform was used to insert a stop codon into the CCR5 gene of human blood cells, successfully conferring resistance to our 5 tropic HIV-1 strains. When compared to CRISPR, the PNA plus PLGA platform offers a safer, highly translatable alternative. Delivery is simpler using FDA-approved nanoparticles rather than complex viral vectors. Because it is purely synthetic, it is non-immunogenic. Most importantly, the structural rigidity of PNAs makes them much less tolerant of base pair mismatches, significantly minimizing genomic damage from off-target effects. We have come a long way from a synthetic curiosity in 1991 to the future of systemic genetic medicine. By combining unrivaled binding affinity with state-of-the-art gamma modifications and PLGA delivery, peptide nucleic acids have overcome decades of biological roadblocks. PNA is now a platform ready to rewrite the rules of gene therapy, oncology, and rare disease treatment. Thank you for your time today. As we move forward, PNAs represent a highly specific, non-viral, and systemically deliverable solution for the most challenging genetic conditions.